Well, I would thank you for the invitation. I'm glad to be participated, and I have been enjoying all these talks in very different topics. So I will be talking about using AFM and molecular dynamics for the studies of the bilayer membrane. Okay, in many biology books, you see this image.
No se ve en la presentación.
And the point here is that, well, this is the Mosaic model, and for a long time it was assumed that this bilayer was just for containing the cell, so there was a sort of inert matrix between the interior and the exterior of the cell. But now it is very clear that the membrane is also involved in very many aspects of biology, in many biological processes. And also that this image of something so tidy, so ordered, is not really true. I will just show, for instance, this is molecular dynamics of a bilayer membrane. And you see, well, it's pretty messy, the membrane, and it can change a lot. Here we are just moving two parameters, the temperature and the amount of esterol. And you see how the membrane gets organized. For instance, for a large amount of cholesterol, this is POPC, a particular phospholipid and cholesterol, and you see how it can pack an order, has an effect of esterol, and also obviously temperature also plays a role. Now, let me give you an example of using the XBio, the microscope to collaborate with people in biology. There are two examples of lymphocytes and how the mechanical properties of the cells affected by two particular proteins here, okay, can have an effect on the behavior of the lymphocytes. So what we did is to determine what morphology and the elasticity of lymphocytes, and this is in the presence or absence of a particular proteins, and how this relate to the process of lymphocyte action. Okay, so this is just getting the nanomechanical properties of the cells, and they have this effect. But we are also interested in trying to understand at a more molecular level how these different processes that occur in the membrane can be understood. Okay, this is Amphotericin B, is a polyene, and is the gold standard for antimycotics. If you get a really serious mycosis, you will have to have Amphotericin, and it's a very potent drug. Is product of Amphotericin in particular is from Streptomyces nodosus. That is a bacteria, so this is the reverse of penicillin. Is what bacteria used to kill the fungi instead of penicillin, and it is very powerful, and the problem is it's also very toxic. And what it does, well, actually there is a lot of discussion for the last 7 decades of what it does, but I would say the idea is that it makes holes in the membrane. This is just another molecular dynamic simulation, and you have the water here, you will have the membrane, and this arranged and makes a hole in the membrane, so kills the fungi cell because it starts losing potassium in particular. And the reason why I can make this sort of supramolecular arrangement is because you have a hydrophilic part of the molecule and a hydrophobic part of the molecule. So it can make a barrel in the membrane, and that barrel makes a aqueous hole where the ions leak. Okay? And if you go to, this is the electrophysiological experiments, and you see how the channel conductivity that you make in the membrane. And the original idea is that esterol, some form of esterol, was needed for the action of these polyenes in general, and Amphotericin in particular. But what we found is that even in absence of esterol, we could have this formation of ion channels. So the idea that you needed an esterol as an integral part of the channel was no longer valid. So we start thinking that probably the effect of esterol was more in the structure of the membrane than in the actual construction of the supramolecular structure. So, and well, this is now more accepted. Now there have been more papers from not only ours, but other groups that are suggesting that the structure of the membrane is affecting the expression of these channels, okay? And here you see, for instance, when you, this is nystatin, actually. This is, and you see if you have a membrane, and this is just simply a bilayer that POPC and just the esterol, ergosterol or cholesterol, and you see how the conductance on the bilayer is increasing. That's because you are making holes in the membrane, okay? And the reason to compare ergosterol and cholesterol, well, because ergosterol is the esterol present in fungal cells, where cholesterol is in the mammalian cell. And that difference, this difference in action is what allows you to use it as a therapeutic drug. Is this a stronger action on the fungal cell, but it's still an action on the mammal cell, and that's why it is considerably toxic. But we did some experiments, and here is one thing that we think it is very clear. If we look to this bilayer and see the phase diagram that occurs when you change cholesterol and temperature, and these are the lines defining the phase diagram between the liquid, disordered liquid, or on the mixed phase and ordered liquid, you see in both cases with ergosterol and cholesterol. And actually these lines are from Prieto's work, okay? So, but then we look to the expression of these channels in this phase diagram, and we see that it's not increasing with increasing amount of esterol. It is more correlated with the phase diagram that you have in both cases. So somehow ergosterol and cholesterol will have different effects on the structure of the bilayer. Well, here you have cholesterol and ergosterol, and there are very similar molecules. You just have a difference. You have a double bond here, you have an extra methyl group, and you have also here in this ring, okay? But they are very similar molecules, but somehow they are able to differentiate the action of polyene. Okay, so let me, one thing that we are doing very much is doing molecular dynamics. I am just giving you an idea of using GROMACS, and we use, for instance, lipids from Stockholm. And well, these are some details of the molecular dynamics. But for instance, if we do molecular dynamics of this POPC membrane with cholesterol, so exactly the same membrane that we use in the experiments, and we change the amount of esterol, and we change the temperature, and we look to the thickness of the membrane. So what we see is that and this is just some artificial pixelation. So we compute the thickness in each of these squares. So you see how the thickness of the membrane is, well, changing, obviously, when you go a larger temperature, for instance, you see how the thickness change. But this is not monotonically, and the membrane is not homogeneous. So there are differences in the membrane structure that we think can be related to the different action of polyen in the membrane. And for instance, if we look to the real distribution function of lipids, and you increase the cholesterol, you see how the packing of the membrane is changing. And something very interesting, if you look to the real distribution function, the real distribution function actually is like sitting on a molecule, or an atom, or on a molecule, and see the surroundings. For instance, this is cholesterols. That means I sit on a cholesterol and see how our cholesterols are rounded. This is the distance from the cholesterol. And you see that cholesterol has a very structured real distribution function. So that means that even if it could look like if it was an homogeneous distribution, it is very structured, and that's because it makes cholesterol makes a lot of lines, or a lot of, shall we say, like filaments of cholesterol that we would see later on. Okay, and also this is the real distribution, the height correlation function. Okay, so what we want to see if the membrane is homogeneous in a statistical manner. Okay, and we see how this is the autocorrelation. This is in time, and this is in distance. And the fact that you get this line here, a bit here, is that you are getting domains. I mean, you are getting parts of the membrane where this correlation of the structures is different. Is that you are getting these domains that were clearer in the images of color, but this is more rigorous statistically. Okay, and here we compare, for instance, the thing that we are very interested in is the main point of this talk: how we validate the simulations. How do we know that we are getting the simulations right? Because there are a lot of approximations in the simulations. So we have to validate the simulation by comparing with some experimental result. And here we have an AFM result on the height. This is in the same bilayer, okay, the POPC and cholesterol, okay, and we have this type of behavior. That means you have a high structure here, and then you go down, and that resembles the thing that you get in the high correlation of the simulation. So this gives us confidence that we are getting is related to expressions in the experiment. Obviously, the times involved here are just nanoseconds or microseconds, and the times involved here are minutes. So we are comparing quite different things, okay? So, but the trend is the same. So it's just to validate the simulation. So we are very interested in this point of validating simulations. Here, I would like to stress something which has been very interesting. Is the Bacterian lecture in the Royal Society in 62 or something like that, see, by Bernal, that he stressed what is a structure in a liquid. And in biology, we have a liquid phase. We have obviously water, and many things occur in water. It's a liquid, a very particular liquid, a very structured liquid. And we have the other great phase, liquid phase, is the lipid in biology, in the bilayers. So, and we have a structure in a liquid. And what has Bernal stressed, a structure in a liquid is a statistical thing. No, it's a statistical concept. I have, for instance, if I, like I mentioned previously, a radial distribution functions of cholesterol that says, or even more better, the radial distribution function of water gives you the information that each water is surrounded by other 4 waters, and that is very similar to the structure of ice. So that means that at the short range, water is like ice, like a small ice. But if you get a snapshot of a simulation of water, you hardly will see for each water like it was an ice surrounding, like the crystal structure. But what you know, if you look the average structure of water, of a single water in time, how, or even in distance, then you get this structure similar to ice with a very, with very short range, 8 angstroms, and that's the liquid structure. So the same thing happened, for instance, in this case, we have, for instance, here the red points are the esterols. So you see there are these lines that give rise to the radial distribution function, these sort of filaments of esterol. But this is the average position for, I think, 30 nanoseconds here. And also, you see, this is, again, the width of the membrane, and you see there are regions which are different. These regions, shall we say, there are high thickness regions, and these are low thickness regions. So there are different regions in this average. And if we look to the radial distribution function of lipids, they are quite different. So that means these ones are more correlated, so these are more structured. Even the density is different because this is a local measure of density, okay? So we have these differences occurring as a liquid structure. And this difference lasts for nanoseconds or more, and so they have impact in what is happening in the surfaces, okay? And for instance, this is the case where you have a more complex bilayer. So you have now esterol, which is yellow, and you have two types of phospholipids. And these are results not from us, from Peter Tillman in a biophysical journal. And these from here to here are 10 microseconds. And this was, shall we say, an homogeneous distribution, and you see how the phospholipids are mixing, yes, going to form domains, and how the esterols are becoming established in the borders, in the interfaces of these domains. So that's the bilayer is an auto-ensemble structure, and all these phenomena occur just in the bilayer. And obviously, when you have biological processes in these different domains, well, they are going to be affected. There are many things that occur in the domains, we'll see a bit later. And here, this is a table that appears in Nature Nanotechnology. And say, when you are using all these very refined microscopies, it's better, as we have seen in several of the talks, even in the previous talk, it is very convenient to combine some of these techniques because they have artifacts, they have limitations, they have advantages. And if you have two of these techniques, then you get more confidence if one and the other are correlated and give you confidence in the results. I add this part, the numerical simulations and molecular dynamics of Monte Carlo simulations, okay? Because actually this would be the ultimate microscope. you get atomic resolution here because you construct molecules with atoms. There are also coarse grain simulations where you make some approximations, and that allows you to go to larger times and larger scales of the systems. But atomistic simulations gives you the high resolution you can imagine because you see where is each atom. But there are also limitations, okay? The result depends very much on the force field we are using, on the intermolecular potentials that you assume for the different molecules. They are limited by the computer time and speed, and obviously you cannot. You have a very refined simulation and an atomistic refined simulation with very refined potential. You cannot go for a very long time, and you cannot have a very large system, okay? So here, what we are doing is trying to see if we can use atomic force microscopy and numerical simulations to mainly to validate the numerical simulation, but also the knowledge that you get from here is going to be extremely useful to understand what you are observing in atomic force microscopy. Okay? And for instance, this is a result. Actually, I would like to mention that some of the details of the experiments present here, they would be presented with more detail by Arturo Galvar in a lecture ahead. So I'm not giving very much of the details, but this is the measurement of height, which is related to the membrane width with atomic force microscopy, and and the coverage of the different domains. But this one is one thing we found is very interesting because the width of the bilayer is affected very with the amount of esterol and the differences between cholesterol and ergosterol. So ergosterol produces a membrane that actually this is a mixture of sphingomyelin and POPC. So sphingomyelin is larger and is a hell at the temperature that we are considering is a hell phase. So you get this width difference. Okay, that are very clear. This is nanometer. It's something like 1 nanometer difference. So we have some observation that characterize the effect that cholesterol, the difference between cholesterol and ergosterol. So we would like to go now, and also doing this, the same thing, the correlation function, the high correlation function, we see that these are applied to the atomic force results. And we see that there are domains, but those domains are different for ergosterol and cholesterol. So we have an observation that we expect to reproduce by the molecular dynamics. But here also, well, to be fair, we have to see if the force fields in the molecular dynamics are adequate. And what we see is, in spite that the chemical structure of ergosterol and cholesterol seem to be very similar. Okay, when you look to, this is a simulation of the motion that the molecules, ergosterol and cholesterol, have in vacuum. It's just simply the freedom that you have for cholesterol and ergosterol. And you see that there is a difference. For instance, here it is the ergosterol is more planar than cholesterol. Okay. And we also, well, for instance, another problem that you have when you do molecular dynamics simulation is you have to compare with something. And even in cholesterol is a lot of information, ergosterol is very little information. But we can go to the quantum mechanical results and say, okay, let me look to cholesterol. and I can do a quantum mechanical refined computation and see how this vibrational surface of the molecules behave, okay? And look to the lowest conformers for cholesterol. And then you look to what is, well, here we have two force fields as lipids and charm for the classical simulation. And you see, well, it's not so bad. Actually, it is very good, the reproduction by these force fields of the quantum mechanical results. When you go to ergosterol and you do the same, okay, and compare to the quantum mechanical calculations, the agreement is not so good, okay? So what you have to do is to refine, you have to go and to refine the potential for ergosterol, and we did refine potential for ergosterol. Say, okay, so now we can go and look to membrane of sphingomyelin. That's here you have the POP. Actually, well, something that I have to mention that is one of the prominence of the molecular dynamics. We saw that in this 10 microsecond simulation, the two phospholipids were starting to unmix, and 10 microseconds, well, is a large time for an atomistic simulation of a membrane patch with enough with a medium number of phospholipids. So it's very costly, time consuming, and in computer time. But then when we did these simulations for the same thing, we do for the atomic force experiment, that's sphingomyelin, ergosterol, cholesterol, and POPC, make a homogeneous mixture and start the simulation and see how the domains will appear just simply because of the separation, because the sphingomyelin is in a half phase. So everything, the thing is that it moves very slow. Even after 10 microns, the domains were not completely formed, okay? So we decided to make something that you have to start to do is to make toy models, okay? And here what we did is, well, better we start from a domain that we construct. So we put the two phospholipids not mixed. We make a sphingomyelin is the green one here, and the POPC is the brown around, and there are some cholesterol there to show. And then do the simulation and see what happened, okay? And we do the simulation. Here we have this image here is for cholesterol and ergosterol, or even if you look, this is just reproducing the simulation box several times in order to get, shall we say, lines like that. So what we have is that, yes, there is a difference in the width, mainly in the height, because we have this part of here is the POPC. This is the width of the POPC phospholipids in the membrane, I would say this brown part. And then you have a difference in height when you get into the sphingomyelin part, okay? So you get this difference between the blue, shall we say, and red regions, showing that the sphingomyelin domain is more ordered, so it is with a larger width, okay? And here we just look into the histograms of these pixels that we define where we compute the width, see? And you see the difference in ergosterol and cholesterol. And here is like doing something similar to the AFM. You can do in AFM experiment and look to the difference in height. See, and you get this. Well, this obviously this repetition is just simply the repetition of the periodical conditions of the simulation. And we get some difference in height, but not the difference that I showed previously of the AFM results that were like a nanometer. And that's a problem because we wanted to validate our simulations, our conditions of simulation. And then we say, oh, we are getting something similar, but not quite, because we don't reproduce this important difference that the AFM was giving, okay? So what we did here, well, obviously we tried many things. We refined the ergosterol potential. We start putting ions in the simulation, different calcium or sodium, trying to say, oh, something is lacking here, and we are not able to reproduce the experimental data. So we are not able to validate the simulation. I have confidence in this system that we have here that we could be using for many other things. But then start looking how it behaves, this simulation. And here you have the local, let me take out this. Well, I hope you see it. I have a message here in my screen, but I hope you see what is the local enrichment of lipids. And that means you look to a lipid, shall we say, a cholesterol or a sphingomyelin, and see how the other lipids are in your surroundings, mainly in the local surroundings. And this idea of how many lipids are around you gives you an idea of how enriched is your lipid in the other lipids or in itself. So they can be by themselves. And for instance, this black line is a sphingomyelin in the cholesterol presence. And we see that as time is moving, and this we have 3 microns here, the sphingomyelin in the presence of cholesterol is getting, well, it was quite enriched because obviously we constructed like that. We make a domain as a starting point, but that domain is losing the domain. The domain is vanishing. Okay, whereas in the ergosterol with the new force field, the domains remain very much the same. Obviously, you would like to have, instead of 3 microns, 30 microns, microseconds to see if this remains like that. That's one of the big problems of the simulation. You don't really know if you are already in equilibrium, okay? But you start seeing the difference here. You say, oh, the sphingomyelin domain is not so stable in cholesterol as it is in ergosterol. Unfortunately, this agrees with the results of the AFM that you will see later on, in that the domains in cholesterol are much smaller. The sphingomyelin domains in cholesterol are not much smaller than those in ergosterol. And also, it seems that here, for instance, this blue line is POPC ergosterol, and this the other one is a sphingomyelin ergosterol. So that means that, for instance, in this case, one is enriched in ergosterol, and the other one is losing ergosterol, whereas here this seems to be distributed equally. So there are some dynamical behaviors that we can compare to the AFM results and see, and they are giving us confidence in the simulation. But we have this big problem that we don't get this reproduction of a large difference in width between the sphingomyelin and POPC produced by the esterols. Ergosterol should order a lot more sphingomyelin, producing a difference in width, and we are not seeing this in the simulations, and that's a really terrible thing, because we have to have this reproduction of the experimental results to have confidence in the simulation. But the advantage is that we can go back and see what happened, why we have this problem. And what we see when we look to the AFM results of the, this is the size of the domains, there are domains of different size, and this is the high correlation, the difference in width between the POPC and the finger, okay? And okay, we can see that cholesterol seems to be lower, and well, it's lower, and ergosterol, there is a larger difference. But we are around here. When we do the numerical simulations, we are in very small domains, simply because we cannot make very large systems in the simulations. And when we are doing the AFM, we are around here. See? So we see and we understand now that we cannot get this very large difference simply because we don't have enough scale in the simulation. But now we are confident that the reproduction that we are giving of this difference, the smaller difference for smaller domains, validate our simulations. And now we can, okay, we need to go to do more time, but then we can see how in this system will a molecule of, or different molecules of different polyen interact with this surface, with this bilayer, with this different, with ergosterol and cholesterol, and start seeing the molecular process that occurs in them, okay? So let me just finish with the conclusions. And we think that joining AFM and molecular dynamics allows very important for validation of MT simulations of biological systems, that in many cases you don't have the possibility of comparing to experiments. And here you produce the experimental observation that you expect to reproduce with the molecular dynamics. Of course, once you get this validation, you can do a lot more comparisons. For instance, the rupture forces that you will see later on, that we measure in AFM, well, those are related to the interaction potential between the lipids and how this potential is affected by the presence of esterols. So we can go to more refined comparisons. But we can also construct simple molecular models, because we cannot simulate whatever we would like. We have limitations in the time, the computer time, and so that's and also in the amount of atoms that we can put in the simulation if we want to have an atomistic description of biological processes. That, as I said, that would be the ultimate microscope. And also, this is, if we get these molecular dynamic results, they will provide information for understanding, for instance, why it is this difference in width produced by ergosterol cholesterol. Was the molecular reason behind this difference, or being more ambitious, going back to the very first slides, why the different proteins have that effect on the mechanical properties of lymphocytes. That's a lot more difficult because you have a lot more things and you have cytoskeleton, and well, but that's looking to the molecular reasons for that differences. Okay, and with this I will finish. Thank you very much.